Hello everyone. In this video, we will explain our project that we made as part of our capstone project, supervised learning techniques to classify cancer at early stage using gene expression data. But before starting to explain our project, I would like to introduce my team members.
Hello, my name is Moksh Mittal. I am in Electronics and Communication, EC1.
Hello, I am Guruansh from Electronics and Communication Department and I am in EC9.
Hello, I am Martand Tewari from ENC3 and I will take you step by step towards the development of the project.
First thing starts with problem identification. Cancer classification using gene expression stands out from the other previous classification that are due to its unique nature and application domain. Through this development, we hope to gain insight into the problem of cancer classification in aid of further developing more effective and efficient cancer classification algorithms. Now, speaking about our project, previously cancer classification has always been a specialist in clinical based terms. Now, with the use of ML and AI, various cancer classification methods can be deployed. But these cancer classification methods are reported to have several limitations in their diagnostic abilities. We soon realized if we can develop some model which can predict cancer and classify them, it would help in the loss of many lives. The recent research of microarray technology has allowed the simultaneous monitoring of thousands of genes, which motivates us to the development in the cancer classification using gene expression data. Now, speaking about the solution part of this project, we basically present a multi-purpose machine learning algorithm to classify cancer at the early stage using gene expression data with higher accuracy. List of genes that differs between two sample sets are often provided by RNA sequence data analysis tool and can be generated manually by statistical testing of datasets. So we try to replicate the classification of cancer using PCA, decision tree, random forest, SVM polynomial, and radial function. Then map the genes using gene expression tools like Panther, Sting, and GeneMania on gene expression pathways. Now, speaking about the flowchart of our project, the most important thing is to collect the data. We collected database from GEO, like Gene Expression Omnius, the TCGA, the Cancer Genome Atlas. Then we pre-process the data, like cleaning of the data, scaling, and labeling of the data was performed. Then we performed principal component analysis, which include the initial clustering, dimensional reduction, and feature selection. Then comes to the model development of our project. We basically splitting of the data, training of our model, and testing of our model was performed. Then after model development, we have to evaluate our model. We evaluated our model on confusion matrices of various model, ROC curves, and the graphical representation of various machine learning models. The result section of our training dataset. The cluster formed using principal component analysis clearly separates the healthy samples from those in the early stage cancer. We got the much higher accuracy during the training of our model. We performed PCA1 and PCA2 on our dataset, and the results came out to be pretty well. Then we calculated the confusion matrix for various machine learning models, and the best accuracy comes out to be in SVM radial and SVM polynomial, which is close to 1. And then we plotted our decision tree, and as you can clearly see, STX11s final. And then we plotted our decision tree, and as you can clearly see, STX11, a tumor suppressor gene, comes out to be the most important feature in decision tree. Then we calculated 10 important features which defines the result of random forest. Then we use gene expression tools like Panther for gene enrichment analysis. Here, there are different processes like regulation of cellular process, biological regulation, regulation of biological process, and development process. These are the processes in which gene is involved, and this is the hit number. How much it is involved in which process, and it is calculated from fold enrichment. Like apoptic process, it is a cell dissection process. Like if there is any problem in the cell suicides, and gene is involved in the process. And if any problem happens in apoptic, then cancer can happen. Like if cell become cancerous and it cannot do apoptic, then it will replicate itself, spreading cancer. Now we have mapped genes proteins on stain. Here we have mapped STX11, a tumor suppressor gene on stain. STX11 is connected to these genes, and these genes are involved in whichever processes STX11 will be directly affected by it. If gene-to-gene interaction is not happening, the gene-protein interaction, whichever takes place, will be affected, and problem will be at the very first stage. Now, here we can clearly see that STX11 is connected to these genes. And if any of these functions stops, then STX11 will be affected and cancer may be caused. Here we have mapped gene interactions on gene mania. Here we have mapped STX11 with different genes. Like STX11 is clearly the most interacting the most with STXBP2 and then with SNAP23. So if any of these genes is affected, then STX11 will also be get affected. Finally, we come to the conclusion that pixel component analysis is a powerful tool for dimension reduction as well as feature selection. The PCA1 and PCA plot clearly shows the clusters between healthy and cancerous cells. The gene selected through the criterion of high loading values shows a clear correlation to cancer as seen in their enrichment analysis in Panther. Web tools like string and gene mania depicting the network of these genes shows certain hub genes which are known as genes in key cancer pathways. Using these as a feature has enabled building of robust ML models that can accurately predict the presence as well as stage of cancer using gene expression data. Finally, our ML model genes with high importance are the major factors in the cause of the cancer, and they have significant roles on the outcome value. And if we can treat these important genes, then we can cure the cancer at the very early stage. Thank you.